Let's begin with the structure of the DNA, under which we will first study the nucleic acid chemistry, under which we will see structure of the nucleotides, structure of the nucleosides, various nitrogen bases that are basically divided into purines and pyrimidines. These are double ring structures, these are single ring structures. Then we will see the Chargaff's rule. We will see the pentose sugar, basically the ribose and deoxyribose sugar. Ribose found in RNA and deoxyribose found in DNA. Then we will study in detail the Watson and Crick's double helix model. We will see the phosphodiester linkage, and in the end we will also see various types of DNA. See, the Watson and Crick model that we study is basically type B DNA, and we will also see the differences between the four types of DNA. Now, the DNA is made up of many nucleotide units, hence known as a polynucleotide chain. Each nucleotide is made up of a nucleoside unit and a phosphate group. These nucleosides consist of a nitrogenous base and a pentose sugar. We can directly say that the nucleotides are made up of three things. First, the nitrogen base, second, the pentose sugar, and third, the phosphate group. Nitrogen base, as earlier discussed, are basically of two types, that are purines and pyrimidines. These double ring structures and these single ring structures. Under purines we have adenine and guanine. Under pyrimidines we have cytosine and thymine. In RNA, this thymine is replaced by uracil. Then we have the pentose sugar. Pentose sugar is basically of two types, ribose found in RNA and deoxyribose found in DNA. Here we have the 5-carbon structure of the pentose sugar. The only difference between these two sugars, ribose and deoxyribose, is the group attached to the second carbon. In ribose case, it is OH group, and in deoxyribose sugar, it is H. Let's see how a polynucleotide chain is found. Here we have the complete structure of a deoxyribose sugar. A nucleotide is formed by attachment of nitrogen base here, either a purine or a pyrimidine, and a phosphate group here at the 5th carbon of the pentose sugar. These nucleotide units are attached by formation of two ester bonds here and here, involving a phosphate group. Thus, it is also known as a phosphodiester linkage. We have taken, for example, adenine as a nitrogen base here. Similarly, thymine here. We have the 5' end near the 5th carbon of the sugar, and the 3' OH end at the 3rd carbon of the pentose sugar. Before moving to the Watson and Crick model, let's see what were the sources for study and analysis of the DNA structure. First was the study of nitrogen-based compositions of sample DNA. Second was the X-ray diffraction studies of DNA. In studies done by Arvind Chargaff, it was seen that the amount of adenine was directly proportional to the amount of thymine present in the sample, and similarly the amount of guanine was directly proportional to the amount of cytosine present in the sample. That is, 8 was equal to T and G was equal to C. Thus, it was concluded that the sum of purines was equal to the sum of pyrimidines present in the sample. That is, adenine plus guanine was equal to thymine plus cytosine. In X-ray diffraction studies, the molecule to be studied is subjected to X-rays. These rays are scattered in a particular way that can be captured on a photographic film. They have a particular pattern. Each molecule has a different pattern. These patterns can be studied to know the shape and structure of the molecule to be studied. Now, the studies done by Rosalind Franklin confirmed that there is a 3.5 angstrom periodicity in the structure of DNA, and secondly that the DNA structure was somewhat helical in shape. Unfortunately, Rosalind Franklin died in 1958 at the age of 37, and since the Nobel is not given posthumously, finally, let's begin with the Watson and Crick model. The DNA double helix model was published in 1953, for which Watson and Crick received Nobel in the year 1962. Some of the salient features of this model are: two polynucleotide chains run anti-parallel to each other. That is, if one chain is in 5' to 3' orientation, the other would be in 3' to 5' orientation. It is a double helix which is coiled around the central axis in right-handed fashion. The nitrogen bases are stocked one above the other. These are separated by a distance of 3.4 angstrom. Nitrogen base of opposite chains are paired together by formation of hydrogen bonds. This takes place here. The fact is, 2 hydrogen bonds are formed between A and T and 3 hydrogen bonds are formed between G and C. We will later discuss this in detail. One complete turn of helix, that is from here to here, 10 base pairs are formed, 10 nitrogen base pairs are formed. Thus the distance between these 10 base pairs or a complete turn is 34 angstrom. The diameter of this double helix is 20 angstrom. And finally the feature that DNA molecule has a large major grooves and has small minor grooves. One interesting fact that when this paper was published in 1953, Watson was a postdoctoral fellow, while Crick, one of the greatest scientists, was just a graduate student in his mid-30s. Summarizing the previously discussed content, we have a portion of DNA double helix here. As we can see, we have a 5-carbon pentose sugar here, from which nucleotides are formed by attachment of a nitrogen base here at the first carbon and a phosphate group here at the 5th carbon. These nucleotide units are connected together with the help of phosphodiester linkage. The opposite chains of a DNA double helix run anti-parallel to each other. That is, if one has 5' to 3' orientation in this fashion, the other would have it in opposite direction, that is, in 3' to 5' direction. The nitrogen bases of opposite chains are linked together with the help of hydrogen bond. If pairing between adenine and thymine takes place, 2 hydrogen bonds are formed. If a pairing between guanine and cytosine takes place, 3 hydrogen bonds are formed. These nitrogen bases are separated by a distance of 3.4 angstrom, and the distance or the diameter of this double helix is 20 angstrom. Lastly, let's compare different forms of DNA. We'll discuss only 3 forms of DNA here: the A form, the B form, and the Z form. A form is formed in concentrated solutions devoid of water. B form DNA is the Watson and Crick model, and Z form of DNA. Now helix, right-handed or left-handed? Imagine if we are moving downwards a spherical staircase and holding both the armrests, and if the central axis is towards my right shoulder, it would be a right-handed double helix. And similarly, imagine if we are moving down a spherical staircase holding both the armrests, and the central axis is towards my left shoulder, it would be a left-handed double helix. So the A form of DNA is right-handed double helix, B form is also right-handed double helix, and Z form is left-handed double helix. The width or diameter of A form DNA is 26 angstrom, B form is 20 angstrom, and 18 angstrom in Z form of DNA. Number of base pairs per turn. That is, in A form these would be 11. In B form it would be exactly 10.5 base pairs per complete turn, and in Z form it is 12 base pairs. Helix rise per base pair. That is the distance between 2 nitrogen base pairs would be 2.6 angstrom in form A DNA, 3.4 angstrom in B form of DNA, and 3.7 angstrom in Z form of DNA.